Let me welcome everybody to the American Diabetes Association Spotlight the Science. This is our third episode in our series where we're highlighting incredible research scientists who have received funding from the American Diabetes Association to advance their work, all with the goal of answering unanswered questions that are about diabetes. And I'm joined today by an incredible scientist, Dr. Hivert. She's an associate professor in the Department of Population Medicine at Harvard Pilgrim Healthcare Institute and Harvard Medical School. And let me tell you just a little bit about the work that she's doing, and you're going to see why I have such a big smile on my face, because we have a great conversation before us. So she is a clinical investigator with a primary focus on the prevention of obesity and related comorbidities, particularly in type 2 diabetes and gestational diabetes. Her interests include fetal metabolic programming mechanisms and the integration of genetics and environmental factors, all contributing to obesity and related disorders. And in her Department of Population Medicine, she is leading research projects that are observational and prospective around pre-birth cohorts and intervention studies in the peri-pregnancy period. That's a lot of information, but how exciting to be joined by you, Dr. Hivert, to talk about pregnant moms, their babies, and how we can ensure health and great health outcomes for both.
Thank you for having me. It's a pleasure to be here today.
So I'm excited to get this conversation started, but first I want to know, how did you get your start in diabetes research?
I would say it started in a couple ways. My first passion for diabetes came even prior medical school, where I was a counselor and a lifeguard in a camp for children with diabetes in California, which was an amazing experience. And at that point, I really found my passion in being a clinician for people with diabetes, and knowing that I wanted to make a difference in people's life by being a clinician that can understand what's really going on with them, and not applying a recipe that for everyone. And then when I was a resident in internal medicine, I was working in the ICU, and it was just striking to me how people in their 30s and in early 40s were coming to the medical or the cardiac ICU and not knowing that they had already type 2 diabetes and other comorbidities, and coming to the hospital with a myocardial infarction at just like 35 years old. And I really felt like I needed to think about a way to prevent those events early on. And so I started working on lifestyle intervention for young adults that didn't have any comorbidities. And then I realized that we needed to think about earlier and earlier and earlier, and then all the way to pregnancy. How can we set people on the right life course of health from the start? And so that's how I decided to start a pre-birth cohort while I was still in Canada as a young investigator. And now I'm working here in Boston with another cohort that started during pregnancy, and that we're now following up both mothers and children, but the children are now young adults themselves. And so we can really think about the whole life course. And in my research, I think about genetics and epigenetics and the biology and the interaction with our environment and our lifestyle, and how to better understand the mechanisms from prenatal through the life course.
Wow. It's so exciting and so interesting how it started with the diabetes camp experience and has led to this fantastic career exploration of all the different life stages. And what strikes me from your story is that now you're engaged in multi-generational projects. I wonder if you'd dive a little deeper and tell us a little more specifics around your ADA research project.
Yeah, absolutely. So I was very fortunate to be supported by one of the ADA Pathways Award to Stop Diabetes as a young investigator. And with the cohort that I just mentioned that I initiated in Sherbrooke, Canada, we had recruited about 1,000 women in early pregnancy, really made sure that they did not have pre-existing diabetes, because we wanted to understand the pathophysiology of gestational diabetes per se, and follow them up throughout pregnancy, make sure to measure both glucose and insulin at multiple time points, including during an oral glucose tolerance test, which is like the wonderful juice that if many of you have been pregnant, you know what it is. And then also made sure to capture all the medical information in a delivery. We collected extra samples on the cord blood that is discarded and the placentas. And now we're following up those families. We completed the 5-year follow-up. We're in the process of the 10-year follow-up. But for my ADA program, what I had the chance to do is to really investigate the epigenetic markers that are present in any of our cell. So we have DNA, which is our code, and our DNA is the same in every single of our cell. And it's slightly different from one individual to another, but not that different. And then around the DNA is how the expression of the gene is regulated, and the markers around the DNA is what we're exploring. And the theory is that there is events during the prenatal development that can impact how those markers around the DNA may be impacted and may have some type of imprinting or programming for the life course. And so that's what we're studying by using the samples we collected in the cord blood and the placenta.
Have you identified a few events or some early findings that you can share with us?
So in the placenta, we've identified quite a few markers that seem to be associated with how the glucose is regulated during pregnancy for the mother. And in the cord blood, we just made a nice collaboration with other cohorts that have collected similar phenotype and samples, and we think we found a marker that also has been found as a DNA methylation, so those marker around the DNA in other cohorts later in life, again arguing for that programming that may be there and present in multiple tissue, in multiple cells, and may impact our life course risk of diabetes.
Wow. So you're working on outcomes both for the mom and for the baby or the child. And as a woman with type 1 diabetes who's had a baby, I couldn't appreciate this anymore. And my heart couldn't be more full than it is right now. So I'm wondering, what's on the horizon? So you've talked a little bit about what you're investigating now, but what's the next? What are you hoping to move towards based on your learning so far?
So on the maternal side, just like you said, so we'll take it one area at a time. The maternal side, we were fortunate enough that based on the interesting discoveries that we've made in the placenta, DNA methylation, so the epigenetic marker, now we have funding from the NIH to look at the next step, which is the transcript from the DNA, so the RNA and then the protein. Because despite pregnancy being a drastic change in how the glucose physiology occurs, and we've known for many years that the insulin resistance or the insulin sensitivity, depending how you want to say it, changes so drastically during pregnancy. And we try to investigate some of the hormones that we know are released by the placenta, but there's still no real explanation that we have how this physiologic change happens. And so this new grant will allow us to look at the whole transcriptomics profile of placenta of women who participate in our cohort and try to identify new protein that are unknown to us that would contribute to understand how both insulin sensitivity, insulin secretion, and gestational diabetes happen during pregnancy.
Is looking at both women with type 1 and type 2 diabetes as well as gestational diabetes, or is it isolated to certain categories?
This is looking at women with normal pregnancies and all the way to the physiology or pathophysiology of gestational diabetes. But we have made sure to not get women who had pre-existing type 1 or type 2 because we wanted to understand the physiology as well.
But I
do care for women with type 1 and type 2 diabetes in my diabetes and pregnancy clinic, and I love working with this population.
It's a special group indeed, and they are filled, especially those with pre-existing diabetes, when they get pregnant, and they're filled with so many fears and concerns and questions about the future. So let's shift. You spoke about the maternal side, but can you talk a little bit about the what's next? What are you hoping for on the infant child side?
Absolutely. So our next step on the epigenetic is to see if really those epigenetic markers are, first of all, if they're present at birth, are they predicting, risk of obesity or diabetes later in life? And that is still to be tested. And the other thing that we're doing is partnering with other cohorts to really confirm our findings, because like a lot of things in science, you need to validate and confirm your findings, even if you make the best and sound investigation, because there are differences in population or other specific things that may be true for your study, but we need to know if it's generalizable. And then the other thing is to get into understanding a little bit more about the appetite regulation when the kids are growing. And so the next phase of our cohort called Gen 3G, we hope to be able to measure how kids regulate their appetite. We're going to do some tests that we call eating in the absence of hunger. And we're running a pilot right now to see if we can do some functional imaging of their brain during a hyperglycemia test. But it's still early, so I don't want to overpromise if that's going to be feasible or not.
That's okay. That's really incredible. And it makes me think we should probably take like a step back and get you to share some statistical information about risk, incidence related to obesity and or type 2 diabetes in the youth of mothers who have gestational diabetes, and that kind of information, just generally.
So you're asking, like, if a woman had gestational diabetes, what's the risk for their child to develop obesity with diabetes in their life course?
Yes.
It varies a little bit from one population to another. And so I don't want to give too many numbers. One study that was done in the Pima Indians quite a while ago by some colleagues showed that in between siblings, if one sibling was exposed to hyperglycemia because their mom had developed type 2 diabetes, which is more than gestational diabetes, but she had one pregnancy without diabetes, and the second pregnancy she had diabetes. And so those are two siblings from the same family, so sharing similar genetic risk,
Yeah.
and probably very similar familial environment. And the difference between the two sibs is about 4 times.
Wow.
To develop type 2 diabetes themselves.
Interesting.
So that's for a woman who developed type 2 diabetes between the 2 pregnancy. But that controls very well for the other risk, which is the genetics and the familial environment. So those are things that we need to think about.
You know, we got a question during our conversation so far from our friends online, and they've asked, how has that changed anything about the research that you're doing in gestational diabetes and the research in your lab?
So I would say for the Gen 3G cohort, we had to postpone when we restarted to see our next wave. We were hoping to start in the summer 2020 to see our 10 to 11-year-old, and so we started only in 2021. The response is really good right now, but so that's one of the impact I would say that has had on our research. On the other hand, we're fortunate that we had already a lot of data and samples collected, so we could still work on that.
It's been a difficult time across the board for people working in every field the last couple of years. But our pathway scientists are dedicated, they're disciplined, they're motivated, they have perseverance like no one else on this planet, and nothing can stop you. And I sense that from just this brief few minutes together. I wonder, since you've had so much exposure over the life course, is there another field that you're interested in? Has this raised more interest for you that you didn't anticipate?
I would say the other cohort that I'm working with here in Boston is called Project Diva, and a lot of the co-investigator and collaborators that are part of Viva have complementary but very different expertise than I have. I've done environmental exposure and science, and social or mental health outcome. So that allows me to learn a lot about other type of science and chemicals like PFAS, and or looking at outcomes like optimism. So that really being here in Boston really allowed me to expand my collaborators and learn about new science that I would not have had the chance if I just had focus on diabetes and obesity. So that's a fun part of being in a big group of collaborators.
That sure is. And that's one of the things that we love so much about the program that you're a part of, the Pathway to Stop Diabetes program, is that there's a large mentorship, collaborative element to that, where the scientists who are the best of the best from around the world intersect with each other and challenge each other and ask big questions, which is really wonderful. As our time is coming to a close, I wonder if you might help us tie a bow on this package of information. What does this mean? All of this research, what does it mean to the person that's watching that has diabetes, or, is the loved one of someone with diabetes?
To me, I think my favorite message is to say, even if I've done a lot of research looking at genetics, that no matter what's your gene or DNA or genetic risk or even familial risk, we are not doomed by our genes. You can modulate your risk and That's what we're trying to understand, is like how to uh, be the understand the mechanism so we can better prevent the diabetes and the complications of diabetes.
And that's what I love about the fact that you're in the Department of Population Health. You're looking at all kinds of factors. And through this discussion, we've talked about prevention, we've talked about genetics, we've talked about some psychosocial and behavioral things, and interests that have come up throughout the course of investigation. And I just need to round it out by saying thank you. Thank you for being dedicated. Thank you for looking at this special population of moms and babies. That's the future. I love it. As a mom of a teenager living through that process with type 1 diabetes, the pregnancy process was unique. It was scary at times, but I learned that it was doable. And the human spirit is powerful. Optimism has meaning, and that women, whether they're at risk for diabetes or not, they're full of potential in this bringing children into the world. So we should never let diabetes stop us from that kind of pursuit, if that's what's in our heart and on our dreams. So thank you, Dr. Hivert, for all of the work that you do. For our professional friends that are viewing this episode of Spotlight the Science, I've got a really great announcement for you. The American Diabetes Association has just opened registration for our 2022 clinical update course, and that will be happening February 4th through the 6th in Tampa, Florida. Happens to be my hometown. And that registration can be found on professional.diabetes.org. We want to encourage all professionals who have an interest in diabetes or touch individuals with diabetes, clinicians, dietitians, educators, pharmacists, social workers, psychologists, everybody is invited to participate in this conference. And this will be utilizing the announcements and then the updates from our standards of care that come out in late December. And so this will be really working towards implementation of those standards. So expect this to be a vibrant, exciting conference. You can find out more at professional.diabetes.org, and you'll see the information there under the tabs that relate to conferences and events. So thank you for joining us. Thank you to our scientists for their incredible work in diabetes. The American Diabetes Association is so proud to support your work and your efforts in our joint quest to answer the unanswered questions in diabetes and to stop diabetes.